Welcome to this virtual lesson exploring gene expression, a topic drawn from Unit 4, Biology. Assumed knowledge for this lesson includes the structure of DNA. If you're not 100% sure about it, please take some time to go back and review that before continuing with this lesson. My name is Charmaine Keel, and I'm a teacher and the head of department at SPARQ Ed. SPARQ Ed is a program run in partnership between the University of Queensland and the Department of Education, which aims to engage and inspire students in STEM and in biomedical research in particular. We are located at the Translational Research Institute at Woolloongabba in Brisbane. By the end of today's lesson, you should be able to define genome and gene, state the central dogma and explain transcription and translation. You should also be able to identify gene regulation factors. The definitions for genome and gene are on the screen in front of you. These definitions come from the QCAA syllabus documents. A genome is defined as all of the genetic material in the chromosomes of an organism, including its genes and DNA sequences. Whereas a gene is defined as a region of DNA that is made up of nucleotides. It is the molecular unit of heredity. Each gene codes for an mRNA product, which is a protein. Remember, a protein is a large molecule that is made up of chains of amino acids joined together. So you can see that a genome consists of all of the genes in an organism, as well as the other DNA sequences that are not coded into proteins. It's good to think of an analogy to help you remember things. A good analogy for genome and gene are a recipe book and individual recipes. A genome is like a recipe book. It's full of individual recipes, but it also has other information. There might be pictures, an index, a contents section that don't actually make food. A recipe is like a gene. It's an actual set of instructions to make a product. This analogy might help you remember the difference. Before we jump into too many details, I wanted to quickly review the structure of DNA and RNA. DNA is known as deoxyribonucleic acid. It is made up of nucleotides. The nucleotides consist of a phosphate, the sugar deoxyribose, and one of four bases, A, G, C, or T. DNA is double-stranded. The double strands run in an anti-parallel nature, so they run in opposite directions to each other, and it forms this double helix structure. In comparison, RNA is ribonucleic acid. It too is made up of nucleotides. The nucleotides have a phosphate, they have a sugar. The sugar in this case is ribose, and one of four bases, adenine, guanine, cytosine, and in RNA, the base thymine is replaced with uracil, represented with the letter U. Uracil will pair with adenine when it's required. You can see that RNA, unlike DNA, is single-stranded. Pause the video here and identify three differences between DNA and RNA. Write them in your notebooks. I'm going to show this slide a couple of times throughout the presentation because it's pretty important. We know a gene is a sequence of nucleotides, and one gene codes for a product. One gene codes for one protein. That is the central dogma of genetics. It's absolutely vital that you understand that. This occurs in two steps. So the gene, which is a sequence of nucleotides, so it's DNA, is transcribed into an mRNA copy. And then that copy is translated in the second step into the actual protein product. We're going to go through those two steps in a fair bit of detail. Before we get to the detail of transcription and translation, let's just look quickly at the structure of genes and the different sections of DNA and what's actually coded and what's not coded. Each gene is enclosed or bookended by untranslated regions. Untranslated means it's not actually, those sections of DNA do not give instructions for the protein. So they're just DNA, they're for other purposes. The gene contains the non-coding introns, which are removed from the RNA copy before being translated into a protein. You can see that happening in the diagram on the right side of the screen. Here's my exon, and when my copy of my DNA is made and my copy is my RNA, you can see there's that exon. In the copy, there's my original intron on the DNA, there's its copy in the RNA. We call this the RNA transcript. It's the first version of the copy. Notice in the next type of RNA that's mentioned, and that's mRNA, M stands for messenger, the introns have been removed. They're spliced. Spliced means cut. So these red sections are cut out of the original RNA copy. So I end up with just the mRNA, which is just the exons. Only these coded exons then go through the translation process to become our protein. There are other non-coding areas on chromosomes. These include the centromere, which is this central section here on the chromosome, and telomeres. Telomeres are the ends. You can see that there's 4 of them and they're circled. There's 1, there's 2, you see what I'm doing, there's 3 and 4. And if I enlarge that, that's a section of DNA, but that DNA is not coded for proteins. So the important information from this slide. There are non-coding sections on chromosomes and even within genes. On the chromosome, some of the sections that are not coded include the centromere and the telomeres, the bits on the end of the chromosomes. Within the genes, bits that aren't coded include your introns. Only the exons are actually translated into the protein. I want to go back to my analogy of a recipe book and a recipe. So remember, a recipe book is like our genome. It's got all of the instructions, all of the DNA. And my recipe is my one list of instructions to give me a product. So it's like a gene, because a gene is one gene gives one protein. So say I wanted to make spaghetti bolognese. I don't need to read an entire recipe book with 50 recipes in it to make spaghetti bolognese. I only need to read the recipe for spaghetti bolognese to make it. So that's kind of what's going to happen here. And more importantly, what it's going to do, because you don't want to wreck your recipe books, in this step, we're going to make a copy of the recipe, so a copy of the DNA that we're interested in for a gene, so then we can make the protein. So this process is called transcription, and transcription is the first step of gene expression. In it, an RNA transcript or copy of the DNA is produced. This whole process occurs inside the nucleus. For this process to start, the enzyme RNA polymerase, so polymerase means makes a long chain of RNA basically, must bind to DNA at the start of the gene to begin transcription. It knows it's the start of a gene because it recognises a particular sequence which is upstream, so just up on either side of the gene. That tells it to start working. So at that point, you'll notice the DNA here has unwound. so it's not in its double helix twisted state. It will unzip, which allows the RNA polymerase to go in. RNA polymerase then builds this RNA transcript. As it opens up, you can see the DNA nucleotide A will pair with its RNA copy or partner, which is U. And at the next point, T pairs with A, etc. And the RNA polymerase will move along and it will glue the individual RNA nucleotides together. It will form those bonds between the sugars and the phosphates on the backbone of the RNA. And it works on this antisense strand of the DNA. I want you to have a look. I'll just erase the lines I put up here so you can have a look. Have a look at how the RNA transcript is similar to the sense strand of the DNA. So because the RNA is complementary to the antisense strand and the sense strand is complementary to the antisense strand, there's going to be a lot of similarities. The only differences will be instead of a T, as you see here, we're going to get a U. But where there's an A, there's an A, because both the RNA transcript and the sense strand are complementary to the antisense strand. RNA polymerase will keep working, will keep building this RNA molecule until it hits another sequence towards the end of the gene, which tells it to stop working. At that point, it will detach from the antisense strand and the DNA will rezip, retwist, and you'll have your RNA copy of your DNA master copy. To check if you've understood that section, let's just do a quick exercise. This is a really common and simple question. A DNA antisense strand has the following sequences of bases. Now, I've organised the bases into groups of 3 called triplets. The question is asking you to write the complementary sequence formed on the mRNA copy. So you can see I've done the first one for you. TTA will form the complementary sequence AAU. There's 3 more codons for you to write. Pause the video and we'll check how you went. The 3 codons you should have written down were CGU, AGC and UCG. So we've just looked at this first step. Remember, our central dogma is 1 gene forms 1 protein. So we've just looked at the process of DNA forming its RNA copy, which is transcription. And you can see that happening here. We have our DNA master copy, it unwinds, unzips, and our RNA copy of the gene is made. Let's look at the next step. The second part of the process is called translation. Translation occurs outside of the nucleus in the cytoplasm, so it occurs at ribosomes. Ribosomes read the mRNA and decode or translate it to make the protein. Let's look at this in a bit of detail. So there's another type of RNA I'm going to introduce you to, and that's tRNA. T stands for transfer. Every type of tRNA has one of these little purple amino acids attached to the end. And different tRNA molecules will carry different amino acids. Now, proteins are chains of amino acids, so you might be starting to see some connections. Down the bottom of the diagram is what's actually happening in the ribosome. So across the bottom, you can see here's my mRNA strand, and we're going to focus on this section and just see what's happening at this one point. My mRNA has the codon UUU. So of course the complementary anticodon, I know lots of names, but that's what it's called, is AAA. And you can see that here on the base of the tRNA. There it is. Attached to that is amino acid, and that amino acid is PHE. So when the codon and the anticodon match, the tRNA will drop its specific amino acid, and that amino acid will join the sequence of amino acids to form a protein. At this next step, you can see the next codon is GGC, so its anticodon is CCG. So it's coming in, the transfer tRNA is coming in, and it's going to drop off its amino acid, GLY. So that will be the next amino acid in this protein. So one tRNA molecule at a time will drop off its amino acids until it reaches the end or the stop of the mRNA. And in that way, proteins are made. So let's just quickly review. So some of the language I've been using, because it does get quite complex. So we have our DNA sequence of 3 on DNA is called a triplet, and an example would be AAA, and it says template there, but remember that's the antisense strand. We've got from there our mRNA codon, so that's the next word that's made, and that would be complementary, so it's UUU. The anticodon is complementary on the tRNA, so it would be the same as the DNA, unless there's a T in there, in which case that would be replaced by the U. And each one of these leads to a specific amino acid, and in this case, it's PHE. Now, I don't remember what amino acids go with what codons and anticodons. You don't have to remember that either. There are tables that you can use to help you work that out. Now, the tables, just be careful, a trick. Most of them are mRNA. See how it says mRNA there? Just check. Sometimes if you look one up online, it might say tRNA or it might say DNA. So you just need to be careful because I'll tell you which piece of information you're going to use to determine your amino acid. Let's look at an example. So if I have my antisense triplet and it was TAC, so the original copy that's going to be made in the nucleus through transcription, my codon would be AUG, T with A with U, C with G. I can then use my table because my table is mRNA, so I don't even have to worry about the tRNA in this example, but I'll just use my table to work it out. Now it says it's going to be met, so let's see where that comes from. So the first base is here, so it was an A, so I go to here. My second base was a U, so there's the second base, so I'm going to be here, and if I go down I can see AUG gives me the amino acid MET. Notice that if it had been any other on the third base, it could have been AUU or AUC or AUA, I would have got exactly the same amino acid. We call that the redundancy of the genetic code. And you can see there's a few examples like over here. The first two letters are important, but it didn't actually matter on the codon what the third letter is because I'm still going to get ARG. That's the redundancy. A whole other topic. I'll let you research that one on your own. So let's practice that. I've given you some DNA triplets. There's 3 of them there. Pause the video. Can you write the codons and work out the amino acid sequence for each? You should now have an understanding of the process of converting the information coded within DNA into proteins. But not every cell has the same proteins active within it or the same proteome, even though they have the same genome. So differential gene expression is what makes cells specialised. And by that we mean differential expression of which proteins are produced is what makes cells specialised. When a functional protein has been produced from a gene, we say it's expressed. So for a gene to be expressed, what steps are involved? Scientists often use different techniques to measure the amount of messenger RNA within a tissue under different conditions to see if those conditions force changes in gene expression. Let's look at the example on the slide. At the start, we have a single cell, undifferentiated animal cell. On the top stream, the neuron-specific or nerve-specific genes are switched on or expressed. So what started as a generic animal cell here becomes a neuron in the presence of those specialist neuron proteins. Alternatively, the second identical daughter cell goes through a different pathway, and under different conditions, the epithelial specific genes are switched on and converted to proteins. The presence of those proteins within the cell cause it to change and become an epithelial cell. All cells within an organism have the same genome, but they have different proteomes. Let's look at some of the factors that can control or regulate gene expression. I'm going to talk briefly through this slide, and then we're going to look at two specific examples. The first way that genes can be regulated, by regulated we mean encouraged to be switched on or expressed or prevented from being switched on or expressed, can be in that before transcription stage. So while it's still in the DNA in the nucleus. Now, transcription factors are caused by interactions with other proteins with a specific DNA sequence. We're going to look at a specific example on the next slide. Another way that it can be regulated within the nucleus is methylation. Methylation is the covalent addition of methyl groups to the base cytosine. This reinforces the suppression of gene expression at the chromatin level because the chromatin in the presence of these methyl groups is more compacted, so it's actually harder to start the transcription process, which remember is unwinding, unzipping, and then the RNA polymerase has to get into the DNA to code it and make a copy of it. With methyl groups, that's more difficult, therefore it reduces the rate of transcription of certain genes. Transcription factors can either increase or decrease the production of messenger RNA, the copy of the DNA. Another type of regulation occurs after transcription or post-transcription. We looked at an example earlier where we saw the splicing of mRNA. We remove the introns and only the exons are expressed. We're going to look at another specific example about how that splicing can be changed in such a way that the final protein is also changed. That's in 2 slides from now. Finally, after translation, so after the mRNA has been converted to a protein, the endoplasmic reticulum or at the Golgi apparatus, different chemicals can be added to the protein which change its shape, and therefore it's going to change how actively it does its job, but also can change where that protein is sent. So that also regulates gene expression by the movement of proteins around the cells. The Lac Operon is an example of a pre-transcription factor, something that's either going to allow transcription to occur or stop it from occurring. This particular example comes from prokaryotes. So here's my strand of DNA. You can see the entire gene is represented by the letters P through A. But the actual DNA to be coded goes from Z through to A. It's this section. These first two sections, P and O, are the strands of DNA or the code of DNA that tells the RNA polymerase chemical to attach and to start working. So that's happened here. Here's my RNA polymerase, it's attached to the DNA and it's reading down the code. It's reading down the DNA sequences, working out what's going on. And you can see it can work and work until it hits a roadblock. And that roadblock is a repressor protein. That repressor protein physically stops the RNA polymerase from moving down the DNA, therefore it stops it making a copy, which prevents the protein from being expressed or the gene from being expressed. I can change the environment these prokaryotes live in to make this gene expressed. If the sugar lactose is added, it attaches to the repressor protein in this location here, which causes it to change shape and it releases from the DNA. Once the repressor protein is out of the way, the polymerase can work the whole way down the piece of DNA. It can make my mRNA, which means I can make my proteins. In this way, you can see how the presence of these transcription factors, in this case a protein, a repressor protein, can either stop the expression of a gene or can allow the expression of the gene in particular environmental conditions. We learnt earlier that not all parts of DNA or all parts of the genome are coded. There are non-coding sections. We had the centromeres between the sister chromatids. That's not coded. The telomeres on the end of your chromatids is not coded. And also sections within the gene called introns are not coded. Let's look at an example of splicing and how splicing after transcription can regulate or control cell function by controlling the shapes of the proteins that are produced. This piece of DNA has 5 exons, numbered 1 to 5. You can see in the RNA transcript, the original copy that's made during transcription, all 5 exons and the introns are copied. The RNA is spliced, so the introns are removed, and 3 different proteins or 3 different mRNAs could actually come about. We're going to look at 2 of them. The correct one is the first one. So here's our mRNAs. So you can see in the first one, I have exon 1, 2, 3, 4, and 5 in the correct order. That mRNA molecule leaves the nucleus, goes to a ribosome and is translated, and it forms protein A. And protein A has a specific shape to do its function. Alternatively, I'm going to go to the third piece of mRNA. This piece has exon 1, 2, 3, and 5. Exon 4 is missing. That piece of mRNA also leaves the nucleus and is translated, and protein C is produced. Protein C is similar to A, but it does have a specific shape difference. You can see there's a section missing around here. This will change its effectiveness, so its function within the cell would be compromised. In this way, the gene expression process is not stopped, but the function at the end can be controlled by changing the protein shape. Protein C would not be as effective as protein A. Look at protein B and have a think about how effective you believe it would be compared to protein A. We're near the end of the lesson, so let's just quickly review our learning goals. You should be able to define a genome or a gene, state the central dogma, 1 gene equals 1 protein, explain the processes of transcription and translation. We're going to do an exercise to check your understanding of those points in just a moment. And finally, you should be able to identify gene regulation factors. To check your understanding of some of the processes discussed in today's lesson, complete the activity on the screen. Match the following words from the word list to each of the indicated structures in the diagram. Pause the video now and have a go at this activity. The answers for that activity are on the screen. M is your DNA double helix. O was pointing to this strand of DNA that's being copied, so it has to be the antisense strand. N is obviously our single strand of mRNA, with a section of 3 bases on that being a codon, which is both Z and X. P is pointing to a spot at the end of a tRNA molecule, so that has to be an amino acid. Q is pointing to the end of a tRNA molecule, but the best match from the word list was tRNA anticodon. And R is your ribosome. Check how you went. You should have got 6 out of 7 or 7 out of 7 for that exercise. If you have any questions or feedback for us, you can contact us at sparked@uq.edu.au. Thank you, and please continue to stay safe.